Today in this class I am explain about the next instruments culture racks. This is the structure of culture racks. Tissue culture racks are very important and essential in maintenance of inoculated culture vessels such as test tubes, conical flasks, petri plates, culture bottles, etc. This rack provides artificial sterilized condition. The culture racks are made up of tubular stainless steel pipes duly painted for long life or made up of aluminium. The size of each shelf is about 48 into 12 inches, covered with thick unbreakable glass which is fired and weather resistant. A fluorescent light are fitted inside the racks. A control panels are fitted at the top of the tissue culture racks. And the next instrument is inverted microscope. This is the structure of inverted microscope. An inverted microscope is a compound microscope in which the objective is below the stage, allowing specimens to be viewed from underneath with first light source and condenser on the top, above the stage pointing down, while the objectives and turret are below the stage pointing up. It was invented in 1850 by J. Lawrence Smith. The stage of an inverted microscope is usually fitted and focus is adjusted by moving the objective lens along a vertical axis to bring it closer to or further from the specimen. The focus mechanism typically has a dual concentric knob for coarse and tine adjustment. Depending on the size of the microscope, 4 to 6 objective lenses of different magnifications may be fitted to a rotating turret known as a nose piece. These microscopes may also be fitted with accessories for fitting still and video cameras, fluorescence illumination, confocal scanning, and many other application of inverted microscope. Inverted microscopes are useful for observing living cells or organisms at the bottom of a large container under more natural conditions than on a glass slide, as in the case with a conventional microscope, and also used in micro manipulation application where space above the specimen is required for manipulator mechanisms and the micro tools they hold, and in metallurgical applications where polished samples can be placed on top of the stage and viewed from underneath using reflecting objectives. And the next instrument is filtration unit, and this is the structure of filtration unit. Filtration is the removal of bacteria by filters. Vacuum filters are used to sterilize solution containing vitamins, hormones, serum, etc., which cannot be sterilized either physical or chemical method. Heat labile fluid like antibiotics, sugars, plant growth hormones, amino acids, and vitamins are sterilized by passing through various types of filters. The filters retain bacteria, and the membrane filters are commonly used in filtration units. The membrane filters are made up of cellulose acetate or polycarbonate that contains pores smaller than bacteria, that is 0.48 microns, and the filtration is usually carried out under negative pressure. The fluid is sucked through the filter into a receiving flask which is connected to an exhaust pump. The filtering apparatus consists of a glass funnel, a membrane filter, a carbon disc, three base rubber stopper, and a flask and a handle. And next instrument is accessories. These are the minor instruments which are most essential and widely used in the tissue culture laboratories. The commonly used instruments are scalpel, forceps, spatula, meme, scissor, surgical blade, cork borer. Scalpel used for excising the explant from plants, and second one surgical blade used for excising and trimming of explant under aseptic conditions, and third one forceps used for holding the plant materials, and the fourth one spatula used for transferring or subculturing the explant or callus, and fifth one is metal cork borer used for cutting the media for antimicrobials analysis, and sixth one is scissor used to fat cutting the unwanted plant materials such as roots and shoots.